Lesson 2 for the chapter Principles of Inheritance and Variation. Let's start this chapter with a short story of Gregor Johann Mendel. Gregor Johann Mendel. He is known as the father of genetics because he was the first to demonstrate the mechanism of transmission of characters from one generation to the other. And this pattern of transmission of characters from one generation to the other, we call it as inheritance. So, we can say that he was one of the person who had given a brief description about the pattern of inheritance by the living organisms, and especially the higher ones. And on the basis of that, the foundation of genetics had evolved. Now let's come to a very short story of Mendel. Mendel was born in Silesian, a village at Hengendorf, now in Czech Republic, on July 22nd in the year 1822 to a farmer's family. He was a brilliant student and studied philosophy for several years. After schooling, Mendel joined an Augustinian monastery of St. Thomas at Brunn, which is now called as Brno in Czechoslovakia, in 1843 at the age of 21. At the age of 25, in the year 1847, he was made a priest in the monastery. In 1851, Mendel was sent to the University of Vienna for study of botany and physics, and he returned to Brunn as teacher of physics and natural science. Mendel served as a teacher for 14 years, and later he was made the abbot of the monastery, and therefore he was given the term Gregor, which was added to his name. In 1856, Mendel observed the occurrence of two types of seeds in the sweet pea plants growing in his monastery. Thereby, he became interested in them. Mendel carried out hybridization experiments on the garden pea for 7 years from 1856 to 1863, and he confirmed the purity of his experimental material through inbreeding. Initially, he took 34 pairs of varieties of pea plants, and later he came to 22 such contrasting pairs, and later he selected only 7 pairs of varieties to study further about the garden pea plant. And in all those experimental methods, or all the selected varieties, he at first selected all the pure lines, or he made pure line breedings, and later he finally came up with the conclusion of his experiments on plant hybridization, which was published in a journal of Proceedings of Brunn Natural Science Society in 1866. Mendel died in 1884 without getting any recognition for his work. But in the year 1901, 3 people, those are Hugo de Vries of Holland, Karl Korenz of Germany, and Erich von Tessermark of Austria, they again revisited his experiments, and they found that the law of segregation and the law of independent assortment that Mendel published in his paper, it had the level of laws of heredity which were generalized by them, and in that way it was again published in a journal called as Flora in 1901, and from then the work of Mendel was established in the study of genetics. Therefore, while studying genetics, we always at first start with the Mendel's experiment. For Mendel's experiment, he had selected his experimental plant as the garden pea, whose scientific name is Pisum sativum, and it bears a chromosome number of 14. That means the diploid chromosome number, that is 14. And the method for his experiment was artificial hybridization. Artificial hybridization means the method of improving the quality of plants by artificial means, which implies artificial pollination, where the pollen grains from a desired quality, it is bought and dusted over the stigma of another variety of the same species. And this whole process, it includes a method which is called as emasculation. So this is briefly called as artificial hybridization. Now let's come to the Mendel's experiment. During his experiment, Mendel adopted 3 steps. The first is the selection of parents, second, hybridization and obtaining of the F1 generation of plants, or we can call that to be the hybrid plants, and the third step is the self-pollination of hybrid plants. Here, a question arises that why Mendel selected pea as his experimental plant. As Mendel was interested in farming and he used to grow several vegetables in his monastery, so once he observed 2 types of seeds in the pea plants that were grown in his monastery. So from then on, he started observing the pea characters and he came up with some characters of the pea plant that were used for the purpose of his experiment. And those are the presence of the several pairs of contrasting characters. The pea plants, some were tall, some were dwarf. That means they have the contrasting pair of characters in their height. Then the pea, they had fertile hybrids. Pea plants, they were bisexual and self-pollinated one. Reproductive organs are well protected, and that makes artificial pollination easier. And most importantly, the life cycle is short, so results can be obtained within a short period of time. So because of such kind of characters of the pea plant, Mendel finally selected the pea plant for his experiment on artificial hybridization. And as said, at first he selected 33 contrasting characters. 34 pairs of contrasting characters. Then he came down to 23 pairs of contrasting character. Then finally he selected 7 pairs of such contrasting characters for his experiment. Now let's come to the contrasting characters that Mendel had chosen for his experiment, which he used as the parents for his artificial hybridization experiment. So these are the characters are the height of the plant, forms of seed, or you can say shape of seed. Third is color of cotyledon. Fourth, color of seed coat. Fifth, color of unripe pods or fruits. Sixth is the forms of unripe pods, and the seventh is the position of the flower. So out of those 34 pairs of contrasting characters, Mendel used only the 7 characters for his study, and he selected those planes for his experiment as the parent. Now, in all of these characters that we can observe, there are two types of contrasting traits or characters that we can say. That means the height of the plant, some were tall and some were dwarf. So tall and dwarf, these are the contrasting characters, where tallness, that is the dominant character, and dwarfness, that is the recessive character. Tallness, it is represented by the Arial capital T, and recessive character, that is dwarfness, it is represented by the letter small t. So as it is in a diploid form, so here we can represent their genotype to be capital T and small t, small t. Similarly, for the other characters also, there are dominant characters and the recessive characters. Such as for forms of seed, there are round seed and wrinkled seed. Round dominant, wrinkled recessive. Color of cotyledon, yellow seeds and green seeds. Yellow dominant and the green color, that is the recessive one. Color of seed coat, so there we find them to be colored, which is the dominant character, and white, that is the recessive character. Color of unripe pods, green, that is the dominant, and yellow, that is the recessive character. Forms of unripe pod, inflated, that is the dominant character, and constricted, that is the recessive character. And the position of flower in the axillary position, that is the dominant character, and terminal position of the flower on the stem, so that is the recessive character. So on this basis, he selected the parents which he had made them pure line. For that, he grew those plants for several generations so that there is no mixed character or mixed gene for his experiment. As we know that in artificial hybridization method, two parents are selected to get a improved variety or to get a desired variety. Now, Mendel adopted two methods for this, or he adopted two types of crosses. One is called as the monohybrid cross and the other is called as the dihybrid cross. Monohybrid cross, that means in order to get the hybrid plant, the cross is done considering only one contrasting character, and on both those parents, which will be the dominant and the recessive one, there only one character will be studied. Say for example, that is the height of the plant. One plant, it will be tall in its height, and another plant, it will be dwarf in its height. So that is the monohybrid cross. And the dihybrid cross is done when two contrasting characters are studied together in that particular plant. Say for example, height and the shape of the seeds or forms of the seed. So there one will be dominant one and the another one will be the recessive one. In that way, he had done the crosses by adopting the artificial hybridization method. Now, what are the steps for artificial hybridization method? The technique of artificial hybridization, it includes 3 steps, that is emasculation, baking and taking, and the third step is crossing over or cross pollination in between the desired variety that was taken in case of the monohybrid or the dihybrid cross experiment. So the technique of emasculation, it is the removal of the male reproductive part of the flower. As we know that the pea flower, that is a bisexual one, and in case of bisexual flowers in the last chapter that we have studied, that was sexual reproduction in flowering plants, that they undergoes or they have the chances to undergo self-pollination. In order to prevent self-pollination, the flower, it is emasculated. Emasculation, very simply, it means the removal of the male reproductive part, that is androecium, from the flower. Now, after removing the male reproductive part, that flower, it is, or the emasculated flower, it is baked and a tag is applied there in order to keep a sign that that particular flower is the emasculated flower, which will include the date of emasculation, the date of baking and tagging. And after that, when the female part, it gets matured enough, as this emasculation process, it is done at the very early stage, during the birth stage. So therefore, as the female reproductive part, it is the gynoecium of the flower, it gets matured, desired variety pollen grains, it is bought and it is dusted over the gynoecium of the emasculated flower and it is baked again. So that is the crossing over or the cross pollination step, which is done according to the desire of the plant breeder, and that is why this technique, it is called as artificial hybridization. So this artificial hybridization method, it yielded a very good result in case of the pea plant, because the hybrids, they were fertile enough to get the next generation, and this next generation plant from these parents, selected parents, they are called as the F1 generation or first filial generation, and the plants, they are regarded as the hybrid plants. So this is the second step of artificial hybridization technique. Lastly, the third step, that includes the self-pollination of the hybrid plants. So when these hybrid plants, they were self-pollinated, the resultant generation, that is the F2 generation or second filial generation, that gave the idea of inheritance pattern in case of the Mendel's experiment. So that we are going to discuss it in the following classes. So in today's class, we just studied about the various steps, reasons that Mendel adapted for his experiment. Thank you.